On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Dr. Tom Bennett from the University of Leeds. And we're going to be talking about a new paper just published in Biology Open, the title of which is Strigolactone Regulates Shoot Development Through a Core Signalling Pathway. So thanks very much, Tom, for joining us today. And it would be great if you can give us a brief overview of the paper, please.
Well, thanks, Brian. Yes, the strigolactone is a relatively recently discovered class of hormone. We've known that they are an internal regulator of plant development for about 8 years now. So there's been a very big push in lots of different laboratories across the world to try and identify the mechanisms for strigolactone signaling.
Okay.
Inevitably, people approach this from very different angles. They're interested in different aspects of strigolactone function. It's happened in quite a few species simultaneously. So a lot of the work was done in rice. Mm-hmm. Quite a lot of it was done in petunia. Some of it in Arabidopsis. And so, really, I suppose this paper was an attempt to nail down nuts and bolts of strigolactone signaling in one species, looking at all the potential components that had been suggested in a range of publications over the last 5 or so years.
Okay.
So we really tried to be as thorough as possible about assessing whether given candidates were really involved. And I could stress this is purely in Arabidopsis, so we try to avoid generalising too much, although we suspect that the pathway we outline is probably quite generalisable.
Yeah. Okay. Well, so Arabidopsis is a model for a good reason, I think. So it's well worth looking at these things in Arabidopsis, obviously. So can you be a little more specific about the findings that you found in the paper?
So the sort of receptor that has been proposed for strigolactones is an alpha-beta hydrolase protein called dwarf14. There are two pretty close homologs in all flowering plants of dwarf14.
Okay.
One of them is completely uncharacterized. And so the first part of the paper is really focused on, do all strigolactone responses really go through dwarf14, or are these other two proteins involved? In a not particularly surprising outcome, we found that it really is just dwarf14.
Okay.
So that's fine.
Mm-hmm.
And then the rest of the paper is really trying to understand then what are the targets of dwarf14 action. So various things have been proposed, including DELA proteins, which are sort of more commonly thought of as targets of gibberellin signaling.
Yeah. Okay.
BES1, which is more commonly thought of as the target of racinosteroid signaling. And then a sort of newly discovered family of chaperone-like proteins, which have the slightly unfortunate name of SMAX1-like, or SMAXL.
Okay.
And essentially, we could show through genetic analyses, some sort of protein-level analyses as well, that really only the SMAXL proteins fitted the description of strigolactone targets. So they displayed quick changes in stability and response to strigolactone addition, and mutating those proteins either suppressed the phenotypes of the strigolactone receptor mutants.
Okay.
So that's kind of strong genetic evidence that they're downstream.
Okay. So you said unfortunately named. So they're called SMAXL because they're suppressors of the MAX2 protein. That's right?
Yeah.
Yes. Right.
MAX2 is an F-box protein, which is part of an SCF ubiquitin ligase. And we've known that MAX2 is involved in strigolactone signaling for longer than really we've known that strigolactones were the compounds that we were looking for.
Okay.
So dwarf14 binds to MAX2. So dwarf14 binds strigolactone.
Okay.
And then binds to MAX2.
Okay.
And through that SCF complex, ubiquitinates target proteins, which turn out to be these SMAXL proteins for degradation.
Yeah, I see. Okay. So the MAX2 protein, so-called MAX because it was part of a family of proteins which were isolated, I think, probably before your time in Ottoline's lab. MAX1 3 and 4, which are also involved in strigolactone signaling, but more in the enzymatic role in the production of strigolactone. So I know a lot of that stuff occurs in the root. So a lot your paper really is focused on the shoot and shoot phenotypes, shoot branching phenotypes. So are you able to say much about what's going on in the root from this data, or is that going to require a whole different set of experiments, or maybe it doesn't have much of an effect there in the root?
So this is a very interesting and ongoing question.
Okay.
So there have been quite a lot of papers published about strigolactone signaling in the roots, mostly though using MAX2 mutants as a sort of proxy for strigolactone deficiency.
Okay.
Unfortunately, there are other signaling pathways that act through MAX2, and those effects of the MAX2 mutant in the root are not all to do with strigolactone deficiency.
Okay.
So if you compare the MAX2 mutant to strigolactone synthesis mutants, it has a much stronger phenotype.
Okay. Okay.
So what we really, or one of the things that I'm hoping to do over the next year or so, is really try and understand what the effects of strigolactone signaling in the root are, and how those that signaling proceeds. Okay,
cool. So hopefully, I haven't let people into your potential grant applications there, but no. So
it's written on my website.
Okay, that's fair enough. No, so I mean, I guess that strikes to one of the interesting things about the F-box proteins in the Arabidopsis themselves. That there's this. I mean, you say a MAX2 has got maybe a couple of pathways which converge into its function, and yet there are 700 F-box proteins in Arabidopsis, and we know very little of what hardly any of them do. So there's certainly a lot of work to be done in that area. So in the paper, you look at the localization of the PIN1 protein, the auxin transport protein. So can you say something about the relationship that you found between strigolactone signaling and auxin signaling in the shoot, just to finish off?
Yeah. So my PhD many years ago was trying to understand how the compound that we now know to be strigolactone regulated shoot branching.
Yeah.
And what we identified at that time was that there seemed that if you looked in the MAX mutants that now turn out to be strigolactone synthesis mutants, there was increased auxin transport. And if you could suppress that increased auxin transport through pharmacological or genetic methods, then you suppress the shoot branching phenotype. So ever since then, we've had this kind of hypothesis that strigolactones regulate auxin transport. And consistent with that, we find that all the known mutants affected in strigolactone signaling increase levels of the PIN1 transporter at the plasma membrane.
Okay.
Similarly, if you mutate the targets of strigolactone signaling, so that if you mutate SMAXL proteins, you lose PIN1 at the plasma membrane. Okay. So you get this nice equal and opposite effect on PIN1. The big mystery that still perplexes us greatly is how strigolactone signaling from the nucleus really has those effects on PIN1 at the plasma membrane.
Okay. Yeah.
So as far as we know, all the core events of strigolactone signaling happen in the nucleus. Within maybe 40 to 50 minutes, you start to see PIN1 come off the plasma membrane in response to strigolactone treatment. So there has to be a link somehow.
Okay.
And that really is a big mystery at the moment.
Okay, so that's obviously something that's going to be interesting to find out in time. So, I mean, the strigolactone signaling module, if you will, isn't quite as advanced as what we know in auxin, perhaps, which have a much larger number of proteins which have been isolated, which respond to auxin.
Absolutely.
So
it's really we have this core pathway that has now been delineated, not just in our paper, but in through lots of publications from a lot of labs. What happens downstream of that, we really are still kind of only just starting to find out. So yeah, we're quite a long way behind the auction field, but that just means that there's plenty of work.
Absolutely, plenty of interesting grant applications to put in, certainly. So okay, thanks very much, Tom. So as for those that don't know, Tom has just started at Leeds University in the last month or so. So just started his group. So we're certainly looking forward to seeing the stuff that he comes out with over the next few years, certainly. So thanks very much for chatting to us about this paper.
That's an absolute pleasure, and thank you for talking to me.